Welcome to the 14th talk of Capstock series. So the last week we covered function theme. This week onwards, we are moving on to medicinal plants and collaborative projects. So today it will be on computational studies of plant stress management. Over to you, Shafiq.
Thank you, Shaila. Good afternoon, everybody. So as Shaila mentioned, for today's CAPS talk, we are starting a new theme, medicinal plants and collaborations. And the first talk is about computational studies of plant stress management. Now, about plant stress management, we have plant often subject to biotic and abiotic stresses, and biotic stress, which caused by living organisms such as bacteria, virus, fungi, pathogen attack, etc. And abiotic stresses are non-living organisms, and those are caused by heat, cold, or salinity, etc. Now, these stresses are affecting plants, and there are some cascade of events happening in the plants to cope up with these stresses. And so here the key events happening in the plants are transcription factor activation and activation of stress responsive genes. Hence, the plant will achieve the tolerance. Okay, so how this transcription activation happens and how the transcription factor will come and bind to the cis-regulatory elements in the upstream of the gene, hence the plant achieve the tolerance. So we will cover all these things in today's topic. So this interest came to our lab through the first grant we received for Professor Saudamni, Welcome Trust Genius Research Fellowship Grant in 2000. And that was the first grant received for our lab. And we studied about structural motifs, sequence searches, and also we purchased our first computer cluster. Now, we started studying about the plant stress management through the second grant department DBT by DBT, and ma'am was co-investigator in that grant. And this this study started by late Professor Uday Kumar from University of Agriculture Science and Professor M.K. Matthew from NCBS. So the project title was to study the transcription factor associated with the desiccation response in plants. Now, for today's topic, we have our first talk about STIFF-DB, that is stress responsive transcription factor database. And this was developed by Dr. Shamir Kader and team, including Dr. Mahandisha and Ambiga, Umman et al. So currently in STIFF-DB, we have 3 plants, Arabidopsis thaliana, Oryza sativa japonica, Oryza sativa indica, and these are the few of the stresses we have listed here. And in the current version of STIFF-DB 2, we have incorporated all these data, and we have incorporated few cis elements, popular cis elements or transcription factor binding site families. Now, Shamir will talk about this project. Over to you, Shamir.
Thank you, Shafi. Thanks, ma'am. Thank you, everyone. Great to be back in the lab. And I have 15 minutes with I hope this works out. Can you guys see my screen?
Yeah, it's visible. We can hear you.
Very good, very good. Okay, thank you, Shafi, for that introduction, and thank you, ma'am, for inviting me to share some of the exciting work that we have been doing over the years. It's a little bit old. I hope I remember everything. The sort of the scientific title for this session, or my part of the talk, is that to delineate molecular players mediating stress response pathways in plants using a digital biology platform. But it's also this project, or rather the series of projects, was also a critical learning piece for me as a JRF in the lab and eventually a graduate student. And this also, I wanted to kind of, when I'm giving you the details about the science aspect, I also want to tell you that when you are a grad student, I'm sure many of you either are or planning to go into grad school, So sometimes the side projects that you work, they'll become so important as you grow into a scientific career. And with respect to the way I see STIF is, I call it STIF suite, because you have algorithm, you have database, you have analysis, you have gene discovery. So you have the whole suite of innovation and discovery in one project. But it just started as a collaboration, if I remember correctly, like maybe Ambika wanted some help with the database, and then it kind of went into a sort of a long-term collaboration, and that led to all of this that you will see today. Okay, with that background, Shafi covered this a little bit, but I just want to, why working on plants and why working on understanding the foundational aspects of plants using a computational approach is critical, because there are several issues, several challenges with agriculture. We are rapidly converting agrarian lands to other forms of development, and we definitely need a balance between the growing human population or the capacity to produce food. There are certain challenges, and food security, just like other global development goals recommended by WHO or other global organization. Food security is a significant issue. And what happens with the crop productivity is that crop productivity is not really going up. There are several challenges, and this is one of the reasons that we were also interested in, apart from the things that Shafi mentioned as well. And in a way, plants are at constant risk of a variety of stress. This could be environmental stress with biotic agents, could be by non-biotic agents, but in the end, ultimately, this stress will affect the plant biology and ultimately the crop productivity, which will in turn help influence the way we have food security as well. And then understanding the pathways, the genes or the drivers of a variety of stress mechanisms or stress tolerance is very critical, so that once you understand what are the genes that are protecting a plant from stress, or what are the genes that are driving or activating stress pathway, if you have that understanding, you could potentially engineer things in a way that to make the plants less stress tolerant. And in a way, we were talking about a couple of years ago, like, especially with CRISPR and other types of approaches were not that popular. But we knew that, like, there are literature reports and resources that are available, transcription factors, a few transcription factors are available. And then what we were interested was, can we somehow identify this transcriptional programming or sort of regulatory network that are driving under stress tolerance? And what we did in this is, maybe like being a computational biology lab back then, is there a way we could potentially look for a sort of a post-genomic solution? This is the time when a lot of these databases have been started getting a lot of data, whether it's sequencing data, whether it's ChIP-seq data, whether it's function annotation of genes. These were all just coming in. And with this background, we started the work maybe in 2007 or so. And we got this first concept of developing an algorithm to predict transcription factor binding sites in plant genome. And then when we had the package ready, we thought maybe we should apply this to a one or two plant model system, and that led to the development of STIFDB. And then eventually Mahantesh joined the group, and then he expanded that to a few other genomes as well. So it's been it was just it started as a small side project, and as you can see that it went over several years, and we continue to do some work in that space as well, which I'll show later today. And the real aspect of this, and for me, was one of those projects that's about building foundational computing capabilities. And this actually, like, being part of this project over the years, really helped me solve a lot of problems within healthcare and drug discovery eventually in my career. So it was a great learning experience. I should say that. And if you are grad students, you should say no to side projects. That's one of the message I wanted to give now. Okay, to start with, the first part of the STIF suite or STIF platform is that it's all started with an algorithm. It's a simple concept. It's still, it was using Hidden Markov model. Back then, it was like almost at the borderline of machine learning, if you ask me. And then the idea really was to look for, we have a set of approved transcription factor binding site, we were interested to use this hidden Markov model, and we can scan the UTRs in both forward and reverse direction and see if there are matching or high probability transcription factor binding sites. So that was the concept. And it uses some aspects including jackknifing models and all. And then we wanted to have a way to visualize this. It was if I remember correctly, this was probably C script, if I remember. And it'll give you this probabilistic score. And we wanted to have a way to visualize this. And as you can see that the screenshot is from, I don't know how many of you use Biopal anymore, but this was, back then, this was the coolest technology. You get the genome, you could find the position, map it. This was all done using BioPython, which we launched Biopal, not Python even. And we come up with the algorithm. We benchmark this against 2 experimentally validated genes. It was picking up nicely with this 90% sensitivity and 57% specificity as well. The algorithm was developed, and we then thought to apply this to around 2,269 genes. But the interesting aspect of these genes were coming from gene expression database. And on top of that, we had some constraints, like we were only looking upregulated genes. And we started with StiffDB1, and with one species. We started with Arabidopsis thaliana, and then we went to develop StiffDB2. So in StiffDB1, we had 8 stresses plus 1 plant, but when we came to StiffDB2, we had basically both more stresses as well as different types of plants as well. And those who are interested in bioinformatics application architecture, this is the standard architecture that we use for StiffDB. But then when we went into the StiffDB2, we had access to more data. As you can see here, for each different plants, we had different number of transcription factors, different number of signals, and data was curated from a combination of literature and gene expression omnibus. And that part alone is, in a way, now as industry standard, if you go to any pharma or agriculture or other large-scale companies, as you can see, that mining data from PubMed and Geo is a pretty standard and in a way, a high-value data mining activity that we all do on a regular basis. And as you can see, 2 types of rice, and then one on Arabidopsis thaliana. And then we applied Stiff algorithm. We identified, we added a few additional tooling around the data as well. And these are the studies we had. And as you can see that, what are these transcription families, or what are these members? As you can see, this includes MIB, BZIP, ERE, BP. So we basically take each of this family and then go into individual transcription factors, find their binding motif, and then do the searching. So it was quite an exhaustive process, but we were able to automate the entire process. Was all able to capture pretty good representation of the stress regulum. So I also, eventually I thought this was also a way to do ChIP-seq without actually having ChIP-seq in there as well. So it was very powerful that way. And that powerful aspect is something we realized, I basically realized much later, because an interesting thing happened, not just about learning from StiffDB project, but we published, Stiff algorithm is published, and then after, like in 2012 or so, we published StiffDB. And a lot of plant biologists were using this to validate their regulatory network in their favorite plant species. So we realized that there's a group of scientists that could use the discovery that we have coming from our computational algorithm. They could use that to do some validation experiments. So that was quite motivating, and obviously led to StiffDB2. And StiffDB2 continued that sort of citation as well. And I think Mahantesha counted at least 40 to 50 different plant species or plant biology labs used to do novel regulatory network discovery using the data from StiffDB. So it was quite interesting, the kind of impact that your work could make across the understanding the plant stress biology. So these are different stresses and then different transcription factors that we have looked across. And then what is the value of this data that we are collected over time? As you can see here, we not only looked at stress regulatory aspect, but as you can see here, we looked at GC content of these stress genes, so we found some difference between them. So like that, we had some fundamental shift in the way we were looking at plant stress biology as well. And then we not only did that type of basic analysis, we did some network analysis of these genes. We also looked at an approach called transcription factor cascades. Whether there is whether there are sort of typically you will have a transcription factor. You'll have the we do the alternative splicing, you get an RNA product, but then that could ultimately, when you have after the alternative splicing, this could encode another transcription factor. So you could have this type of cascade that happening, and this has a huge implication in stress biology, and then also interestingly in human disease as well. But then we were looking at this aspect from this database as well, and we looked for at least we identified around transcription regulatory cascade for at least 19 transcription factors from the data set as well. This was also a purely interesting text mining exercise that we did. And in a way, one of these genes, like in this ethylene responsive transcription factor, and it had a predictor transcription factor for abiotic, and then we were able to find that they were important part of a cascade, and this was again a piece of discovery we had, which eventually validated us. So we also did some network analysis. So cascades was one aspect. So we also did some network analysis. We identified certain key sort of driver genes within these transcription factor, stress responsive networks. And as you can see, these were some of the key functions of that. And this one, interestingly, we eventually published this as a preprint. So apart from the manuscripts and other things came out, we also did a preprint publication from STIFF portfolio. And as you can see, this data was so rich with information that's not really been explored. So we developed an analytic framework, and we did some analysis. We tried to understand for each stress, what are the functions that are driving, what are the protein domains that are driving, what are the plant phenotypes that are associated with stress responsive gene, and we gained a lot of interesting information from that as well. So this is our meta-analysis framework we have used for this as well. And not only that, the Mahantesh and Shafi has extended the work to do some sequence and structure prediction as well. Okay, so we did some gene ontology analysis. Obviously, we cannot do a bioinformatics investigation with that. There's some beautiful plots here about the function. These are some of the key protein domains that were associated with it. We looked for enriched protein domains within each stress data sets. As you can see here, these are the different stresses and different protein domains that we have. Yeah. Okay, so before I finish this part, there's this interesting part we did was when we were at this, we were doing this work, we wanted to do some planned phenotype enrichment analysis, because a lot of Go terms and whether it's biological process, molecular function, these were all sort of overall for biology in a broad sense, right? Like, so, but we want to go a little bit more specific and want to understand what is the implication of these genes in the setting of plant phenotype. So we did some analysis and we realized that maybe a tool to do this type of work might be interesting to broader audience, and we released a tool for that. That's called POIS. We call it POIS. It's called Plant Ontology Enrichment Analysis. And again, this work was also published and also still available from CABS. I checked, I think, a few months ago. Yeah, so and what do we get here? We get to know the genes and their associated phenotypes. For example, are there genes associated with leaf production that are implicated in biotic or abiotic stress? What are they? So that type of information, which we don't really get from Go terms, that's where the value of plant ontology enrichment came. And there are two sort of term space. These are different terms enriched in these genes, as you can see here. And again, we also looked at, did a final analysis, sort of a final analysis, like are there master regulatory genes that are driven, potentially activated by all type of stresses? And there was a couple of these genes, and one of these were 81 G68440 was responsive to 7 different signals as well, including ABA, drought, heat, light, osmotic stress, and wounding as well. And then we looked at their, the function. We did some modeling of these proteins as well. So as you can see, it all started with a simple concept to do a a software to do transcription factor binding sites, but it kind of went all the way into understand some of the foundational and fundamental aspects of plant stress biology. We also, we continued that collaboration, and this was something that we did in 2018, 19 maybe. We had an invited review and wrote a very extensive review about this whole aspect. It's also in a way motivated by what was happening in the healthcare space. Like healthcare was moving more towards digital health, and as you can see here, we thought, with the availability of low-cost sensors and the way that we are collecting more data, both molecular and non-molecular data, we can get real-time insights from the farm to laboratories, and this could ultimately improve crop plant varieties, because a lot of sort of the foundational data could come from resources like StiffDB, but then a lot of data that we could read from fields as well. So you could ultimately do what we call as digital farming that could in turn lead to sustainable agricultural practices as well. So I encourage you to read this review as well, if you are interested in that type of approach. With that, I would like to conclude the session. We developed an algorithm. We developed 2 iterations of a database. It's an important resource for plant biologists who are studying plant stress. We looked at the functional landscape of that stress responsive genes. We identified some novel genes as well. And we have extended our collaboration in very many ways. GKVK is where it all started, but then we also tried to do some other collaboration as well. With that, I would like to thank ma'am for all the opportunities, and Mahatesh, Ambiga, and all the lab members, and of course Susan and Professor Dekumar were our collaborators from GKVK. This was funded by CSIR, and that's also something that I'm really proud of, because this was funded back in 2005, 2006, right? Like someone thought this idea have so much value, and I cannot thank whoever approved this project. Actually, it was quite an exciting one for me personally as well. Yeah. With that, back to you, Shafi. Thank you.
Thank you, Shamir. Very well, nicely explained for both StiffDB, Stiff algorithm, POS. You covered everything. Okay, please stay on. We will have question and answer session at the end, and we are following to a discussion. Okay, so let me share the slides again. So we heard from Shamir, and next we move to the second talk from today's second topic from today's talk. We have Dr. Pankaj Bara. He was associated to the lab during his MS thesis research. Later on, he moved to NTNU for his doctoral studies. So he will talk about plant responses to single and combined stresses. So he has looked at several ecotypes from Arabidopsis thaliana collected from throughout the world, and he has looked at single and combined stresses for Arabidopsis thaliana, and he collaborated during his doctoral studies with the core team of StiffDB, and he has done some Stiff analysis to study his combined stress. So we will hear from Dr. Pankaj Bara regarding this work. Please, Pankaj Bara, you may share the screen.
Yeah, hello. Good afternoon, ma'am, and good afternoon to all of you. So thank you for introducing the topic, and thanks to Samir for introducing the Stiff algorithm. I just start with some basic concept. We know the plants, when many people have been studying plant stress responses in the lab condition, and why it is important to study plant stress already all of you know, because of the food security and climate change. But when we look what is happening in the field, most of the plants are exposed to multiple stress at the same time. But when most of the stress experiments done in the laboratory, they consider one stress at a time. So during the course of evolution, we know the informations are stored in the genome, and the information get decoded, transcriptome, proteome, metabolome, and finally we have all these interactomes, gene regulatory network, protein-protein interactions, metabolic pathways, and all are modular in organization. And then what we get is basically the phenotype. And nowadays also the phenomics, high throughput phenomics field is developing. And there are different kinds of interactions. It is biotic interactions, biotic abiotic interactions. So the plants are exposed to many kinds of biotic and abiotic factor. And there always been kind of, there is a selection pressure to get adapt and evolve. And through this, the entire plant and stress interaction can be studied from both top down and bottom up. So something is happening in the phenotypic level, and it is introducing some diversity in the plant, which is getting introduced in the genomic level. On the other hand, we know that there are spontaneous mutations, spontaneous variations, which are taking place in the genome. And then due to the selection process, they are getting incorporated and we are getting phenotype. So from that aspect, it can also be studied from the bottom-up approach. Now, because of these kind of continuous interactions, as we know that plants are sessile, they cannot move from one place to another place. They have adapted with robust stress response mechanism. Gradually, they develop stress tolerance. Then some of those species or some of those ecotypes or cultivars, they develop stress resistance. And in the due course of adaptation, the genome evolves. This is an example of the model plant Arabidopsis thaliana, which is actually found all across the globe, starting from the Alaska region to desert to high altitude. So, there are so far around more than 3,000 different ecotypes of this model plant Arabidopsis thaliana available. So, immediate question was how this Arabidopsis thaliana species is adapting to different climatic conditions. And you know that this is a model organism, and understanding a genetic or molecular basis of how a plant adapts to different climatic conditions is very essential in today's date to accurately predict what is going to happen in future during the course of climate change. So this is one of the examples how climate change is going to affect agriculture. This is one picture which is showing about the rice productivity, the severity of loss of rice productivity in Asia by 2050. And as I said, that the stress in plants are not occurring alone in the natural conditions. There are so many different combinations. So how do a plant react to when multiple types of stress are given to a plant? This was not studied well. For example, some of those interactions can be positive, some of those interactions can be negative, some of those interactions can be neutral. This was some very early hypothetical work, review published by Ron Mittler in 2006 about some combinatorial effect of some abiotic stress, right? Now, what is so far known about how do plants react to any kind of stresses, biotic or abiotic? If we just think in a mechanistic point of view, the plant perceives a stress signal, and those signal input is transduced by different molecular signal transduction mechanism like MAP kinase, ROS accumulation, hormonal plant phytohormonal signaling. Then the signal crosstalk takes place. So if it is a single signal or a single stress, so there is like maybe a more clear picture of how the signal is getting transduced. But if more than one kind of stress comes together, then how those signals will crosstalk with each other. There are multiple players like the transcription factors, which actually play a crucial role during this signal crosstalk and signal processing mechanism. And then there are a lot of, even in the transcriptional, translational level, we have a lot of complicated regulatory mechanisms, post-transcriptional, post-translational mechanisms. And in the end, we get the signal output in terms of differentially expressed genes or differentially expressed transcriptome. And then finally, the signal response takes place. Now, this is actually some of the partially known picture of how different signal crosstalk when multiple biotic and abiotic stresses are given together to a plant species. To understand the complexity of combined stress, there was an international consortium initiated by a few people in Europe. With the name of that consortium was ARANet Plant Genomic Consortium, where they considered 30 different plant species, including some of those nature. So the blue ones are basically natural ecotypes of the Arabidopsis thaliana, and there are single stresses and combination of stresses. So there are 6 single stresses: highlight, heat, cold, salt, and flagelline. So flagelline actually mimics the biotic stress, and there are 6 other combinations. So total 11 different single and combination of stresses. So they have been given to all these are basically the mutants. These are mutants. The red genotypes are mutants. So they have been given to all these genotypes, and they profile the transcriptome and metabolome from all these experiments. So it was done way back in 2007 to 2011. And I joined Professor Atle Bones' lab as a PhD student, and I got the opportunity to take over the entire data, and it was a great opportunity to visit most of these labs and spend time to understand how they are doing the experiment to get the data. The most striking question that we wanted to understand was how the plant perceived the combination of stresses, how the response of the plant to combination of stresses, and can we understand any kind of mechanistic view how the plants will respond to any new kind of combinatorial stresses. So this was the experimental setup for, I mean, it was a very large scale experiment conducted for the first time in a very uniform setup. It was done in the Rio National Laboratory in Technical University of Denmark in Copenhagen. So all the plants were grown in a same condition to reduce any kind of experimental bias. So these were the normal plant growth conditions. And then each of the plants were given treatment for 3 hours. And all the experiments were conducted on plants of 3 weeks old. And we have generated the transcriptomic data set, and these data sets were submitted to NCBI GEO with this record. Here I give you some example. These are basically the ecotypes which were collected from different latitude, from 16° north latitude to this 51.5 north to 16 degrees south. It's from Cape Verde Island, Kashmir, India, up to Belgium. And when actually we look at all these 11 different type of stresses, so these are basically the single stresses in yellow, and here in the green you see these are combination of stresses, and these are the numbers which we identified as differentially expressed genes in that specific condition, in that specific ecotype. So if you see the number of differentially expressed genes within the same ecotype for different stress conditions, this was different, and for the same stress conditions for different ecotypes, it is different. So it already shows that the plant response to a same stress condition among different ecotypes is different. And that already gives us some hints about the differences in the transcriptional regulatory network among all the different ecotypes. So similarly, here you see this is one ecotype, and how the number of differentially expressed genes in response to different ecotypes also, because Colombia is one of those very standard ecotype that people use to do all stress experiments in Arabidopsis thaliana. So here actually one thing was clear, that when you use a specific ecotype for doing any kind of stress experiment, the results that you get, if you use some other ecotype, it may differ. So that is why I think it was a very interesting message that we have also passed on, that when you do any kind of stress experiment, the selection of the genotype makes a crucial decision on that. Just to make the story short, what we did, we have finally clustered all the differentially expressed transcripts into 5 different categories. We call combinatorial, cancel, prioritize, independent, and similar. So what we have found, that these combinatorial, cancel, and prioritize, these are the categories of the transcript for which the expression signature of the differentially expressed transcripts could not be predicted just by looking at the expression signature of the single stress conditions. Hence, we have said that the transcript level response signature in plants to combination of stresses would not be predicted just by looking at the expression signature of the single stress conditions. So this is another example from the same data set where we have shown that using one stress condition, which is cold, there are differences among different ecotypes, how they respond to the temperature stress. So this is showing 1,000 top cold regulated genes in all the 10 ecotypes of the Arabidopsis thaliana. Then we have actually looked into the cold stress responsive regulon, that is called CBF regulon. And here also we have shown that there are significant variations in the expression signature, and as well as there are significant changes, variations in the sequence level variations within the sequence level analysis. And we have done some evolutionary analysis to show that these kind of core stress responsive regulons were under positive selection pressure. Then we looked into the differentially expressed transcription factors, and we found that 294 stress regulated transcription factors were there in all the 11 stress conditions. So this shows that these are all different 11 stress conditions, and this shows how many transcription factors are overlapping with multiple stress conditions. So here you see those are the transcription factors which are overlapping or shared in multiple different stress conditions. The number of transcription factors which were differentially expressed in multiple stress conditions were very less, whereas the number of transcription factors which were specific to a very specific stress or at least 2 or 3 stresses were more, that you can actually see in these peripheral numbers. So these are the numbers. So finally, we grouped all these differentially expressed 294 stress regulated transcription factors into 39 different transcription factor families based on the consensus motif based on the published literature. So as we have got some idea that the gene expression difference was mainly because of this differential regulation of the transcriptional regulatory network, then as because I knew that Mahante Saab, ma'am, and Samir was involved in this stiff algorithm, so we actually have utilized some network component analysis. That is one very standard algorithm using our published microarray data in all these 10 different ecotypes, and we have extracted the transcription factor binding site from all different publicly available database for Arabidopsis thaliana, where we have got around 3,500 differentially expressed genes and 294 different transcription factors. We have generated a transcription factor and target gene interaction network using network component analysis. And then we also have predicted the activity of the transcription factors based on the expression profile of the genes and the transcription factors. And then we wanted to validate this kind of co-regulatory network which we have constructed using the computational model. So we have collected data from ATHAMAP, ARANET, and literature survey. And then additionally, we thought of comparing, because the steep algorithm was quite powerful, so we wanted to compare the prediction of the transcriptional regulatory network by generating another transcriptional regulatory network from the steep DB. That is how we have collaborated with MAM's lab again, and we have actually generated this transcription factor target gene interaction network. So this network shows the overall topology of the first ever, I think, this kind of a transcriptional regulatory network that we have constructed for the model plant Arabidopsis thaliana using 11 different stress conditions, that to single and combination of stresses. And so just to do that, our network was not random, so we have done random network analysis, and so the random network, our network was actually scale free in nature. And this is actually one of the example of the sub network. So we had 11 different stress conditions. So we have created the global network, and also we have extracted the sub network. We have identified individual hub genes within the network. As some of those, like cold stress, heat stress, those were very quite well studied stress. So we have already validated that we were able to pick up some of those very well known transcription factors for those single stress conditions. So hence, what we actually propose, that those transcription factor hubs that we predicted for combined stress conditions would also be important, master regulator for some of those stress regulatory network in Arabidopsis thaliana. Again, so this is basically the gene set enrichment analysis of our regulatory network and differentially expressed genes, which again, actually, highlighted some of those stress related processes, which are quite well known. Primary metabolism was compromised, the secondary metabolism and the stress response processes were basically, more positively regulated. So then we have identified the cluster of transcription factor binding site. So the logic is that there is called homotypic cluster of transcription factor binding site. So in most of those interesting, very master regulators of stress like ME, BH, LH, BZ transcription factors, which are known as versatile master regulator of stress responses, what we have observed that there were multiple sites for the transcription factor binding site. And why we thought that it might be important for conserving the robustness of transcriptional robustness of the versatile master regulators, so that even if some binding sites are disrupted because of mutations or any other event, still that versatility of the core stress response mechanism of the plant will still be intact. So to sum up, for the first time, we have shown that the plant's stress response to combined stress conditions could not be predicted just by looking at single stress conditions. We have identified a couple of potential combined stress responsive genes, and then we have generated a stress regulatory network in Arabidopsis thaliana using 11 different conditions and 10 different ecotypes. We have identified a couple of combined stress regulators in the model plant Arabidopsis thaliana, and we have shown how the plant actually regulates the processes to cope up with stress, the core stress responsive genes, and the secondary, more transient and very specific stress responsive genes or transcription factors. We have also shown that homotypic clusters of transcription factor binding site may play a very crucial role for maintaining the transcriptional robustness against any kind of cis regulatory mutations. So this was the work that I did, and I came back to India in 2017. Now I am working in Tezpur Central University in Assam. So after coming here, I again started working. So earlier I was working on the model plant Arabidopsis thaliana. I wanted to do some more translational work. So here I have now initiated some experiment on rice. So these are some of the experiment that we are still working on. We have recently developed a knowledge base on combined stress in knowledge base in crop plants. So we have 11 different crops, 13 different crops, and 11 different combined stress conditions. So I think in future, if we like do more collaborations for combining stiff DB and these kind of resources, so we'll be able to do more productive and more interesting stuff in combined stress responses in plants. So with this, I would like to thank all the collaborators, including MEM and the funding agencies which have supported during this part of the work. Thank you very much.
Thank you, Dr. Pankaj. It's a very nice talk. Please stay on. So we heard from Dr. Pankaj about single and combined stress. Now we will move to the third story from today's talk, that is sodium transporters in rice species. Now, this work started from Dr. Gayatri Venkataraman from MSSRF Chennai. She, after her postdoctoral research, she continued the work, and currently she is at faculty at MSSRF, and she came up with some interesting questions regarding salinity tolerance in rice species. So she approached ma'am, and we have done very nice structural work. And this was started around 2016, where Ritvik was involved, a postdoctoral fellow. He was involved in homology modeling of these rice transporters, and I myself continued the work later on. So I'll be talking about this work in the next few slides. Okay, so Orisa species, there are 27 different species. They are from 11 genome types. as 6 are diploid genotypes and 5 are polyploid genotypes. Now, salinity stress in rice, we know salinity stress affect crops worldwide, and cultivated rice, that is Oryza sativa, that is highly sensitive for salinity stress, and it will affect in both seedling and reproductive stages. And this salinity stress mostly due to the excess of sodium ions accumulation in the shoots of this plant. Now, it has been reported, or it has been discovered, that this HKT gene family members, they play a central role in controlling sodium ion accumulation in the plant. That leads to the conferring salinity tolerance. Now, HKT here, HKT here, that regulates the central role, which will transport sodium ions from root tissue to the shoots. Here, this is a simple structure for the HKT transporters. Here we can see 4 repeated transmembrane P-loop transmembrane domains, and in each P-loop we have selectivity filters, and based on the selectivity filters, it will decide whether it is sodium ions or potassium ions to transport. So here in this rice transporter, we have in the first selectivity filter position serine, which means that will transport only sodium. So if you have glycine here, it will transport both sodium and potassium. Okay, so Ritvik and ma'am started modeling this HKT protein. HKT protein, as we know, this is a membrane protein and it's very dynamic. There are no crystal structure available, and we selected Oryza coctata, an allotetraploid plant. This is a very saline tolerant plant and found growing in coastal region of India and Bangladesh. Now, so with collaboration with Dr. Gayatri Venkataraman, we have done some kind of experiments. You can see here, this is a cultivated rice species and Oryza coctata, the halophytic species, where when we provide 80 millimolar sodium chloride, and now Oryza coctata is showing the saline tolerance here, and where Oryza sativa is very saline sensitive. Now, for modeling, what we have selected 2 bacterial crystal structure. These are not HKT proteins, but they are potassium channel proteins. So we use the information from these 2. Basically, we use this as templates. Both structures are template, and we model them. Now, the core structure, the main template we used as 3BJZ. The N and C terminal are mostly similar to 4J7C. Here, these are the model. This is done by Ritvik and ma'am. And ma'am is to tell this is the highly challenging work, where the percentage identity between these 2 templates with our sequence was around 20% only. So we had to do a lot of loop refinement and loop modeling to get a good model out of it. And following to this, we have done molecular dynamic simulations, and through the structure, we identified certain amino acids changes between these 2 species. And here, what you see here, Oryza sativa HKT protein, and this is a specific variety Nippon Bare, which is very highly sensitive. And this is Oryza coctata HKT15, that is a very saline tolerant species. Now, I'll show the molecular dynamic simulation video where you can see the important residues around the pore region where the ion pass through for Oryza sativa gene and Oryza coctata gene. What you can see here, these 3 gene here, they are repelling each other because they are same charge residues here. Here in the pore region, we have a positive charge residue here, and because of that, they are having a salt bridge formation, and it is attracting to each other. Okay, so we have done some transport kinetic assay studies and conductivity studies with the help of a lab from France. And we saw this OCHKT15 from saline adapted Orisa species and got a lower affinity to the sodium transport. When we are comparing to the Orisa sativa HKT transporters, that has a negatively charged amino acid residue at the same position as the Orisa coctata, but it conferring 16-fold higher affinity transport. So that was surprising. A halophytic species like Orisa coctata having a lower affinity compared to a sensitive one. Now that leads to a further study. So the next study what we have done, we selected subgenome type of Orisa coctata. So it's allotetraploid KKLL. The first one we have studied was OCHKT15K, and we have taken a homologous OCHKT15L. And if we see the alignment with the template, you can see both Orisa coctata genes are mostly similar, at least at the C and N-terminal end. And there are few differences, and with the template I have shown this in this alignment. And if you closely look at the alignment to the K and L, here there are few differences we have shown here. These are the black color asterisks are like 2 nanometer from the pore region. What are the changes in the residues are? And there are other crucial amino acid changes what we can see here, and I'll show that in the model. So we modeled them just like how we modeled the OCHKT 1.5 OCHKT 15K. Now here what you can see when you compare here, there are a swapping of residues. Here you can see in the K protein we have a aspartic acid and a lysine here. Okay, here instead of aspartic acid we have arginine, here we have a glutamic acid here. A swapping of the charges happened here when we compared between the K and L proteins. And we validated this model, and we could find like there is 93% of the residues are in allowed region. Also we validated with the PROSA. And we applied electrostatic map to this both protein. And so here what you can infer, here is the pore region, and just over the pore region we have a positive charged residue lysine here. So we can expect like when a positive charge residue like sodium, when it comes to the pore region, because of this positive charge residue it will repel. So that's why the affinity is very low for this OCHKT15K compared to the Orisa sativa sensitive one. And when it comes to the Orisa coctata HKT15L, you can see a negative charged residue just next to the pore region. And there is a swapping of the charges happen between these crucial residues. And also we can see a positive charge patch here, which may direct the sodium ions to the pore. Okay, so to summarize, we in the earlier study we have compared with Orisa sativa HKT15 and with OCHKT15K. And here we saw some salt bridge formation between these oppositely charged residues, and which gave higher conductivity for the ions. But compared to the Orisa sativa HKT15, which have negatively charged residue around the pore region, it has lower affinity compared to the Orisa sativa. Now the new homolog we studied from Orisa coctata itself, we can see a swapping of the charges for around the pore region, and we can expect like there is a salt bridge formation here and also salt bridge formation here, and it's just like Orisa coctata K protein. Now it since the pore region, the mouth of the pore region have this glutamic acid as a negative charge residue that can attract the ion, sodium ion, and also it can have higher conductivity just like the K. So this work was done between Professor Saudangmi from NCBS, Dr. Gayatri Venkataraman from SSRF Chennai, and Professor R. and Alinor Vedi from Montpellier, France. And the first modeling was done by Ritwik. And these are the funding for this project. So currently this project is continuing by the funding of CEPRIPRA. Okay, so for today's talk, we have covered 3 topics, and I would request Vikas to share relevant publications for this today's talk. Vikas, please.
Relevant references from today's talks. So the first one is about the stick algorithm, and from Sundar et al. And then the next 2 are related to the databases developed using stick algorithm, ScriptDB and ScriptDB2. And the last one is related to another server called POES for the plant genomic analysis and enrichment analysis server. Next, we also heard from Pankaj about the combined stress. This is the publication related to that work. And lastly, we heard about the sodium transporter from the Oriza plant from Somasundaram et al. And from Pulikapti et al. Next week, we will be gazing into some interesting medicinal plants through the lens of genomics. So stay tuned.
Thank you, Vikas. So coming back to my slide. Okay. Okay, so for today's talk, we had 3 speakers, and first one was Dr. Shamir Kader, talked about STIP algorithm and STIP database. He was the one of most efficient, ma'am used to say, PhD student she got, and he had worked a number of other databases and web servers, not only STIP and POES. And currently he is executive director in Sanofi, United States. And the second speaker, Dr. Pankaj Bara, he's currently assistant professor at Tezpur University, Assam. And myself, I'm a postdoctoral fellow, continuing the work I just explained. Okay, so that's all for today. So as Vikas mentioned, we will see the next week for the second topic.